Hello. I think there were some questions on November 13th at the PSOC meeting in Princeton about fixation time and population size, so I wanted to give you some intuition about what a fixation time is. Let's consider a model system involving two types of cells, black and white. And this icon for a black cell is larger to indicate that there's a selective advantage for it. Let's consider oops, it fell on the floor. Let's consider the population dynamics for an example system consisting of 3 cells. At generation 0, there are, let's say, 2 white cells and 1 black one. And then we can represent that inside this plastic container, 2 white cells, 1 black one, and use this container to talk about reproduction. This is Wright-Fisher reproduction, a simple popular model. Shake the box, and you randomly take out a cell, and it happens to be white. In this case, put it back. Okay. Shake the container again, pick out a cell. Oh, it happens to be black. Put it back. Shake the container again for the final cell, which happens to be black again, and then take that new population. What did we say? We said white, black, black. That's generation 1. You could take generation 1 and put it inside the plastic container and then shake and draw cells out and continue. And ... later, which I will not demonstrate right now, we could have, for example, 3 black cells. Because the selective advantage is for the black cells. And once you get all 3 cells being black, the plastic container will only contain black cells, which means you'll never pull out white cells, and the color doesn't change. The color black has, so to speak, fixed in the population. Great. Let's call this whole sheet of paper going on to infinity time for a set of three particular cells, slots, I guess, that you put cells in. This is an experiment, and we could do repeats of this experiment with lots of other sheets of paper. So what are some of the properties of those experiments? Well, quite a few of them will end up having all 3 cells be black at some point, just because it is an advantage. But those experiments won't necessarily turn all black at the same time. There's some random variability associated with the process of the shaking of the plastic container and the taking out of the cells. So maybe on this one, it was generation 5 when all of the cells turned black. Forever turned black. But maybe in another experiment you have to wait till generation 10, and maybe in another experiment it happened really fast, maybe even on generation 2. Okay, so that's one thing to remember. There's not, if you set up an ensemble of these experiments, you're not necessarily going to have fixation happen at precisely the same time. Okay, and you're not going to have fixation necessarily happen in the same direction. You know, there's only 3 slots here for cells. 3 cells we're talking about at any one time. So there is some chance that I could have an experiment where I pull out 3 cells in one generation. Oops, they were all white. And that means that the population actually fixes white. White fixes in the population. So I want to say again that fixation, turning all one color, doesn't necessarily happen at predictably exactly the same time every time you do this experiment. Okay, and it doesn't necessarily happen, end up turning into the same color in every experiment. All of these caveats aside, you can still say on average, when you expect the colors in various populations to stop changing. Whether that means that could mean that they all turned white or that they all turned black, but there's some average-ish time when you say that the colors for many experiments, put a whole bunch of these next to each other, after which, you know, most of the colors stop changing. And that's called the fixation time. Yeah. So now let's get to the intuition about population sizes. You're starting the fixation time that I think we were discussing in the discussion on November 13th is is related to a particular example where you start with one mutant out of a population of other normal or wild-type cells, whatever reference cells. And the question is, how long does that color take? How long does it take for an experiment starting from one mutant plus everyone else normal to get to the point where they're either all normal forever, or they're all mutants, for instance, all mutants forever, basically. I think I just lied and I left out a special case where I guess there is a possibility that a small set of these experiments could perpetually be mixed heterogeneous, but I'm going to ignore that case for this discussion, or else it'll get very long because these digressions are... Anyway, yeah, so if you, here's an intuition. If you start the population with, let's say, 3 cells, there's one black cell, and I guess there's not that many other cells for this black genotype to have to work its way into. There's only 2 other slots left for it to dominate. So you expect maybe it doesn't take that many of these drawing processes for that to happen. Now let's consider a very big population with like 1 million cells, only one of which is black, and it still the selective advantages have for the black cell is still the same as it was in here. It might take a really long time in terms of numbers of generations, how many times you have to select 1 million, draw out 1 million of these manipulatives from the plastic box that magically contains 1 million manipulatives too. It might take a lot of those cycles before even you get a significant number of the 1 million cells turning black. And you might have to wait a very long time before they all turn black, if they do. Intuition. Fixation time becomes long with large population size.